Hi, this is Professor Himmelblau, and welcome to the latest Bio 161 pre-lab video. This one focuses on the Photosynthesis Lab. Of course, you're going to want to read the lab manual, and also in the lab report booklet, you're going to need to do your pre-lab. So photosynthesis can be thought of in two separate reactions, the light reaction and the Calvin cycle. So both take place in the chloroplast. In the light reaction, light provides the energy to create excited electrons. Those excited electrons then give off their energy, and it gets stored in the form of ATP and NADPH, a reduced molecule that stores energy. Then also inside the chloroplast is the Calvin cycle, and that's where the energy in ATP and NADPH is used to take carbon dioxide out of the atmosphere and reduce it. And those reduced carbons are what make sugar, like glucose. So an important connection between the two is that the ATP and the NADPH that's made during the light reaction, that's what provides the energy for the Calvin cycle. Now together these make up photosynthesis, but in this lab we are only focusing on the light reaction and just thinking about those excited electrons as they move through the membranes of the chloroplast. So this is a thylakoid membrane in the chloroplast, and you should recognize some of the proteins that are embedded in here. There's the photosystem II and photosystem I, and then a number of other electron carriers. And remember, a big part of what's going on here is that NADP plus is going to be reduced by electrons. So when the sun is shining, what happens is electrons become excited and they start flowing through this pathway, and eventually they get deposited on NADPH. The first thing you're going to do in this lab is isolate chloroplast, and you do that by grinding up spinach leaves. Now, the procedure is pretty straightforward, and your TA will review it with you, but you're going to grind up the leaves in a buffer. You're going to filter those leaves to get big chunks out. And then finally, you're going to spin them in a centrifuge. Alright, so this is my sample that just came out of the centrifuge, and if you see, you can look up here at the top, you can see that some of the material is collected down at the bottom. This is called the pellet, and the liquid up above it is called the supernatant. Now I need to separate those, and that's not very hard to do. That pellet is tight enough in there that all I need to do is pour off the supernatant into a new tube, and you can see that the pellet is still stuck there. It's okay, I didn't get all of the liquid off, but most of it's off and the pellet is still there. Now, this pellet might contain my chloroplast, so I want to put it on ice right away. And now this supernatant that I've collected, this goes back into the centrifuge for another spin. Alright, so this is that first supernatant after it's been spun again at a higher speed. And you can see that now there's a new pellet down here at the bottom as well. So again, I have to separate the pellet from the supernatant. And when you're finished with this, you'll have 3 tubes, and I've already got mine labeled here. We've got P1 for pellet number 1, pellet number 2, that's the pellet produced in the second spin, and then finally S2 supernatant number 2. Now one of these 3 is going to contain our chloroplast that we'll be using for the rest of the experiment. But before we check for that, we have to resuspend these pellets, which means to break them up and get them back in solution. And this is a step where a lot of people go wrong because they don't do this well enough. Spending a little extra time at this point resuspending these pellets can really save you a lot of time down the road when you start analyzing your chloroplast. So you're going to need to mix this with 1.5 mils of this grinding buffer. So you can just use these pipetters. They're marked here with the amounts of milliliters on there. So I'm going to put 1.5 mils of that in there. Now the pellet still needs to get broken up, so what I'm going to do now is reach in here and just kind of squish it up and down. Stop every now and then and check to see if I have any chunks, and if I still see some chunks, I'm going to keep squishing it till all those chunks are broken up. So really look carefully and take your time at this point, because it's going to make everything work better later on. Now once it's done, once it's resuspended, I want to definitely put that right back on ice so I can protect any chloroplast that might be in there. Of course, you're going to do the same thing with pellet number 2. So I compared all 3 of those solutions using a microscope, and I found out which one contained the highest number of free chloroplasts, and it was this one here. So now what I need to do is make one more solution. This is called my Chloroplast Dilution, and this is really important because this is what we're going to use in all of the subsequent steps of the experiment. Now I've already got 5 mils of the grinding buffer in here, and then I'm going to add to that 1.5 mils of this chloroplast containing solution here. There's 1, and a half. Now this I'm done with. I can put this away, and this chloroplast dilution is what I'm going to use in the rest of the experiments. So definitely I'm going to want to leave this on ice to keep the chloroplast happy. We like happy chloroplasts, don't we? In this week's lab, we want to watch the light reaction taking place, and to do that we're going to use a molecule called DCIP. So DCIP is blue when it's in its oxidized form. That's why it's shown blue here. Now it can also pick up an electron, and then it will become reduced. And in its reduced form, the DCIP molecule is clear. So if there's electrons around, the DCIP is going to go from blue to clear, and we can watch that happening using the spectrophotometer. So where do the electrons come from to reduce DCIP? Well, in this lab, we're going to mix the DCIP with the chloroplast that we identify in lab. And when the lights go on, then the electrons that are moving through the system are going to get stolen by the DCIP. It's an electron stealer. And so it's going to take those electrons, and that's what's going to reduce the DCIP and cause it to become clear. And we can watch that happening with the spectrophotometer. Now we're ready to do the first activity, and this is one you have to think through very carefully before you begin. So I've already got the spectrophotometer set, and I have blanked it. The other thing I have set is a tube that has some of the DCIP solution in it that we're going to use to detect the presence of electrons. Now, you can see this tube fits inside this little cassette here. This is going to be important because when we're ready to go, I have my light bulb here, and I'm going to hold this right up against the light bulb, and this is going to establish the right distance between the tube and the light to give me the reaction that I need. The last thing I've got is my chloroplast dilution ready to go. So now that everything's set, I can take my blank out. I'm going to add 1/2 ml of my chloroplast dilution to this. Quickly, I'm going to wipe this off and get my initial spectrophotometer reading. Now once that's been done, I'm going to take it off, cover it over, put it in the cassette, and then hold it right up against the light. And now I'm going to wait for 1 minute. Okay, 1 minute's almost up, so I'm going to take this down, pick it up, mix it quickly, wipe off any fingerprints, and get a spectrophotometer reading. You have to, don't wait for this to stop because it's going to be changing as it goes. Once you have the spec reading, this goes back in the chamber and back up at the light for another minute. And you're going to take readings every minute for 10 minutes. So now you've finished taking readings with the spectrophotometer every minute for 10 minutes, and you've recorded those in your lab notebook. Now you need to graph those values, and we provide a graph for you, but it doesn't have the y-axis labeled. You need to look at your numbers and figure out what's going to make the most sense for labeling your y-axis. Then when that's done, you go ahead and you plot out your graph. Alright, now to figure out the standard reaction time, what you're more or less going to be calculating is how long it took for about half of the DCIP in your sample to become reduced. And that's going to be a function of your highest absorbance reading and your lowest absorbance reading. Now we're not going to go through all the calculations here, they're in your lab manual. But more or less you're going to figure out the midpoint between those and then draw over from the y-axis till it intersects your curve and then draw down, and that's going to give you your standard reaction time. In the case of my sample, it's been about 4 minutes. So for all the remaining activities, I just need to put my chlorophyll sample in the light for 4 minutes and then take my final reading. So now you've extracted chloroplast and you figured out the standard reaction time for your sample. Now it's time to test the activity of that sample in different colored lights. You'll do white and black, of course, but in addition you'll also do red, yellow, green, and blue light. Another thing that you'll be testing is the effect of DCMU. DCMU is another electron stealer, just like the DCIP, and you're going to see how it affects the ability to detect those electrons moving. And finally, this whole thing gets a lot easier at this point, because now when you put your chloroplast solution and your DCIP into this little box, you don't have to hold it there in front of the light for 10 minutes. You only have to hold it there for the standard reaction time. Whatever you calculated for your sample, that's as long as you need to wait.